Alrighty, so we are now focusing in on a different aspect of biochemistry in terms of structure, and protein structure is probably going to be one of the most important things that we are going to look at for a majority of what we see in terms of function. And so we're going to see that proteins form a lot of structure and a lot of functional items, but the concepts we learn in this chapter will be always zeroed back to give the reasons why things happen. Okay, so there's some fundamental things about the ideas that we learned here. Also, when we learn biochemistry, this is what we do. All right, there are thousands of individual proteins that work. We're going to give you examples of about 6 or 7 that you know in very good detail. From those 6 and 7, you need to know them in detail in and of themselves. But what is more important is that you extract principles that you see in these that you can apply to the other systems. And so this is how biochemistry is taught. We can't teach you all 1,000 different proteins that are working, but we give you examples of certain classes by which they work. Then when you do research or you look at something specific, then you have a framework by things how things generally work, and then you look at the differences or specifics about the one thing that you're looking at. Okay? And so we're going to give you some examples, some typical examples of things that are always studied. Not only are they always studied, they're the ones that we know in most detail. Not only are the ones that we know the most in detail, they're the ones that are tested on standardized exams. Okay, so you will see one protein's function that we're going to look at. We're going to compare hemoglobin and myoglobin. Okay, it is one of the most studied proteins that we have. There's a lot of detail that we know about it, and so you'll see it quite often on standardized exams, so knowing about that. Also the exam that I give. And so we'll take a look at some very typical proteins. Protein structure and function. Okay, proteins, they are a polymer of monomers called amino acids. Okay, and there are 20 standard amino acids by which proteins are synthesized from. The proteins are synthesized only from monomers in amino acids that are of the L version. Okay, there is L and D. They could be classified R and S, but we use L and D and for the terminology here. All right. Amino acids are very important because they have different sizes, shapes, and the properties that they bring in terms of hydrophobicity, how hydrophobic it is, its polar, whether it's charged or not. And these side chains are what's going to give the protein function. Now, the average size of a protein is somewhere around 110, the average size of an amino acid is about 110 what they call Daltons, okay, which is another terminology for grams per mole. So it's a molecular weight. Okay, so each amino acid is about 110 on average. All right. Again, they're larger, they're smaller, but that's a good average. Here is an amino acid. The reason why it is designated D or L, R or S, is because it is chiral for many of them. Not all of them, but many of them. The general sense, amino acid, because it's got an amino and an acid group on it. Wow, that's really hard. And has a proton, and then R. All right, and so the R's are what's going to differentiate the amino acids, and you're going to need to know a couple things. Okay, you're going to need to know the structures. You're going to need to know them well enough to draw them. Okay, so drawing all 20 amino acids, important. It's important to be able to draw all 20 amino acids. It is important to be able to draw. Okay, and the R groups are what's going to be important. Now, the R groups then, not only do we need to know them individually, but we need to be able to classify them. because groups of amino acids tend to provide function, okay, as kind of a group. If it's one of these, it gives this function. Or if it's one of these, it gives this function. And so we kind of group them together. So here is a group. Nonpolar aliphatic R groups. Okay? So the types of interactions that we have with these are generally only the van der Waals forces. We call them hydrophobic interactions. They're relatively weak. They're mainly carbons and hydrogens. And so aliphatic, they hate water. They love to interact among themselves. And so the only one that is not really that aliphatic is glycine itself. This is the one that is not doesn't have a stereoisomer because it has 2 protons. Even though methionine has a sulfur in it, because it is bound in a thioester, it is nonpolar. All right. Polar uncharged groups. Okay. So these are groups that tend to like to interact with water. Okay. They tend to be ones that are accessing the cytosol. They have the potential, many of these have the potential to make strong hydrogen bonds. Not all of them, but many of them. And some are reactive. The cysteine is reactive. And so they will be grouped and they will behave very similarly within the protein, where it's placed inside the protein and what kind of function it has. Aromatic groups. Okay, these are particularly important in terms of, if you classify them, 2 of them are very hydrophobic and really kind of associate in that aliphatic region. There's one tyrosine that has a hydrophobic portion and a hydrophilic portion. You have the hydroxide there. What's important about these in particular is that it gives a special property to proteins. We can actually measure the concentration of protein in a solution based upon these aromatic rings, because we can place it into an instrument and we can take a measurement. Okay? Does anybody know what that instrument is? What? It is part spectroscopy, but what kind of spectroscopy? What is it? Oh, yeah, we can put it in infrared, but that's not what an organic chemist would do. That biochemist wouldn't. What is it? Mass spec? We could do mass spec, and we do mass spec for protein sequencing, but not necessarily. That would be for all amino acids, not just aromatics. NMR. NMR, again, that is, we do analysis on proteins, but not particularly just for aromatics. But we'll talk about NMR. That's a good thing we'll look at. Okay? It is because of the conjugated double bonds. All right? Conjugated double bonds tend to absorb energies. If you get enough conjugated double bonds, it absorbs in the visible range. Okay? There's not enough conjugations here to absorb in the visible range. But there are enough conjugated double bonds to absorb in the UV range. So UV vis spectroscopy. Okay, what we'll do is we'll place a solution inside a UV vis. We will shine UV light into the sample. The sample will absorb UV light, and the amount of absorbance is dependent upon how many things are there, how many of these amino acids are there. And based upon the protein, you can actually calculate the amount of concentration based upon how much absorbance is there, and you can estimate that concentration without losing any sample. It's actually pretty powerful. And so we like these because they have a unique property about them. Negatively charged R groups. Okay. Here is a problem when we look at this. It is negatively charged. Yes. Okay. But it also has a positive charge and another negative charge. What we're talking about is the negatively charged group on the R group. Okay. And so The functional group that we're showing is what in the R group? What is that functional group? Carboxylic acid in the deprotonated form. Is that the only way that you can find a carboxylic acid? Mm-mm. It can be protonated, right? If it's protonated, what charge is it?
Neutral.
What is it?
Neutral.
Neutral, right. And so, but I classify this as negatively charged. The reason why I classify this as negatively charged is this. The pKa of the side group, okay, and the pH of the solution, normally, between the two, the solution is so basic compared to the acidic nature of those that they're always deprotonated under physiological conditions. So if this side group is exposed to the bloodstream or the cytosol, the pH of the solution will always deprotonate them. Okay? And so they are almost exclusively found as negatively charged if exposed to the cytosol. Their protonation state can change if it's on the inside of a protein, because inside the protein makes a microenvironment that's a little bit different from the outside. Okay? And so when you see this, it should be drawn as negatively charged if it's normal conditions. Okay? Positively charged residues. Got 3 of them. All right? Now, can anybody call me a liar when I say this?
Histidine.
What is it?
Histidine.
Yes, histidine. What's going on with histidine? I said this was positively charged R groups. Lysine you can see, arginine you can see, right? They're always charged. Histidine is right on the edge. The histidine's pKa is very close to the pKa or the pH of the solution. And so very often the histidines are charged, but sometimes they are not charged, and really what's really happening is in a protein the pH and the pKa are so close that is a percentage charged and a percentage not charged. We'll look and see where that charge actually comes in just a moment, but histidine tends to be charged in a lot of different functional aspects of biological function. Yes?
So would we draw it like this?
Ah.
Conditions, or do we draw both?
You can draw either. What you would have to do, and I would accept either. What you would have to do, unless the pH and the pKa are absolutely defined, then you would have to draw it based upon rules I'll teach you in just a moment about that. So you've seen 20 amino acids. What do you need to know about them? You need to know what? How to draw them. Okay. Important. All right. Now the amino acids, a lot of them have long names. Okay, some of them have short names, but we're lazy. So we give them acronyms. And a lot of times you will see 3-letter acronyms, which are pretty easy to identify. All right. But then there are single-letter designates, because really journal articles cost a lot, and they cost by how many pages you use. And so if you want to give the sequence of an amino acid, actually writing all their names out is too long. It costs you money. Even doing 3-letter designates sometimes is too much. So they go down to single letters because it's cheaper. Okay? And so which one do you have to know? Yes. Okay. Now, here's something that is interesting. You have 20 amino acids, and you have 22 3-letter codes and 22 single-letter codes. What? That's wrong. Okay, here it is. All right, you have glutamine that on the R group has an amide, a ketone plus an amine. Okay. And then you have glutamic acid, which has this as the group. Then you have this letter designate, that's This is GLN for glutamine, GLU for glutamate, GLX for I don't know which one. Okay. Now, there is going to be a experiment that we do, in which case we're going to identify what amino acids are there in a protein. And we do that by breaking peptide bonds and breaking them into individual amino acids. Well, a peptide bond, a bond that bonds two amino acids, is between the nitrogen of one and the acid of the other, and creates this structure right here. So when we break peptide bonds under this experimental condition, we also break any one of these, into a glutamate. And so under those experimental conditions, whether you start as a glutamate or as a glutamine, at the end of the experiment, they all are glutamines. Glutamates. And so I can't tell what it was initially. So that's why we have this extra designate for glutamate, glutamine, also for aspartate and asparagine and aspartic acid. Those two have the same conversion, and so we will see these extra two. I'll show you that in a moment. It's called amino acid composition. Under those experimental conditions, we can't distinguish what they were initially. Okay, and here are the other designates. Most often, we use the three-letter designates in our kind of lectures and things. Every so often, we'll use a single letter, but the ones that are used quite often are repetitive, and I would expect you to know the name and the three-letter code. I would expect you to look up the single-letter code. Now, I told you there are 20 amino acids. That is kind of a lie, okay? Because anything that has a carbon, a nitrogen, an acid, a hydrogen, and something else is an amino acid. There are 20 amino acids used by our molecular mechanism to synthesize proteins. But there are a whole bunch of unusual amino acids. Okay? Most of these unusual amino acids are functionalized after the initial protein is made. Okay? Now, you may have learned back in genetics that the conversion of RNA into protein is translation. And so some proteins get what are called post-translationally modified, and they get modified, and usually they have function. Okay? But this is not something that you can see in the DNA sequence itself. All right? And so there are unusual amino acids, meaning that they're derivatives of some of the side chains that are important for function regulation, and we'll learn about them. So you don't need to memorize these. Know that there are odd ones, and as I teach you new concepts and show you a derivative, that's when you need to know what it is and what it does. At this point in time, just know that there are extras. Lots of them. All sorts of those derivatives. Okay? One derivative that happens and that is fundamental are two cysteines. And this creates a lot of, we introduce this because this has a lot of influence into how proteins are structured. The sulfurs of the side chains, the SH, can react in an oxidative redox reaction to create what is called a disulfide bond. This disulfide bond is very stable, and it is very limiting to the flexibility of a protein. And so disulfide bonds are one of the derivatives of amino acids that is very influential when we talk about its full structure. And so the disulfide bond I wanted to introduce to you at what that is. Ultimately, we said it was a polymer. And so the polymer here has an amino acid 1 and an amino acid 2. They will react together, and water will be the leaving group. Now, unfortunately, the artist really messed this up, okay? Because you will never find a Well, maybe, you might. Well, you'll never find this. You'll never find a solution that has this structure in the deprotonated form and this structure in the protonated form. The pKa's, there's no pKa that will satisfy both of those at the same time. Okay? But it's easy for us to see because, so the artist did this. If I take one of those off and I take that off, that's water. Okay? It's a little bit more complicated than that, but that's why it's shown. All right? And what you can see is a bond is formed between the nitrogen and the carbonyl. In organic chemistry, that highlighted functional group is called an amide. In biochemistry, it's called a peptide bond. Okay. Now, something very important about this structure here. In terms of orientation, the front of an amino acid or of a protein is right here. Okay, we call that the amino terminus. It's the beginning, it's the start. What was the front of DNA? What was the beginning or start of DNA? What side? 5'. Okay. Here, the beginning is the amino acid, the amino side. And over here, the end is the C terminus, or the carboxy end. So the N terminus is the beginning, the C terminus is the end. Okay. Every amino acid is attached to another amino acid through the nitrogen on the carbon and the acid that is on the carbon of the amino acid. What is linked together is the R groups. The information per se, what's translated from the DNA to create the protein, whatever that information was in the DNA, the instructions, is translated into the sequence of the side chains. And so the functionality of a protein is what side chains are linked next to each other and in the entire sequence. So the function will come as a result of the side chains. The side chains will cause the correct three-dimensional fold of the protein and provide the context for what the function of that protein comes about and works at. Now, that particular amide bond has within it a resonance structure. Okay, if I do all sorts of things here and actually write out the Lewis structures, which I know you guys can write out Lewis structures. Whoops. Okay, there is a resonance structure. Stop. Okay, it's not going to not being cooperative here. Okay, in which the electrons can go this way and the electrons can go that way. And so the electrons are resonating back and forth between those bonds. Now, that has a consequence. Normally, a carbon to nitrogen bond, single bond, allows rotation. Single bonds you can rotate. But what happens if you have a double bond between a carbon and nitrogen? Can it rotate? No. All right. I don't have a single bond here. I don't have a double bond here. I have a partial double bond. What does that do to the rotation value around that bond? We know what happens with single. We know what happens with double. What happens with a resonance?
Can it rotate just a little bit?
No, you think it might. But that resonance enough actually gives it enough of the value of a double bond that it doesn't allow rotation. Because it's going so fast and going back and forth that it behaves like a double bond. Okay? And it becomes rigid. What this does is it limits the flexibility of the protein's ability to actually fold. Okay? Now, one thing I did need to talk to you about is on a amino acid, Okay? There's a carbon. That carbon has an R group. That carbon here has an R group, a hydrogen. Then it has an acid side and an amine side. This carbon is called the alpha carbon. Okay, the one in which all four things are attached to it. Okay. When I make a peptide bond, this peptide bond acts like it is rigid, and there is no rotation around this particular bond. And then on this side, there's no rotation. Okay. But along this alpha carbon, in front of it, there is single rotation, and behind it, there is single rotation. All right? So what I've done is this. If I take a string, okay, I can ball up the string in a little tiny ball that's really compact, right? Okay? But that string would represent total freedom of movement, total rotation of freedom everywhere. Okay? All right. The protein doesn't act like a string. The protein acts more like a chain. Okay? And if you have a chain, the links get jammed up. And so when you twist a chain, it's kind of a little harder to do. You can compact a chain into a small ball, but it gets kinked up a little bit because there is resistance to rotation in during every link. It's the same kind of concept here. I have freedom of movement to compact, but it is limited. This helps us out in terms of being able to predict what kind of folds might happen inside a protein. Okay? The values that are looked at are the psi and the phi angle that are in front and behind the alpha carbon, and they are limited. Okay? They're not completely available to us. Okay? Because of this R group right here. The larger the R group, the less freedom you have because of steric hindrance. So glycine has a very large range in which the phi and psi angles work, because the hydrogen is really tiny and that's no problem. But a tryptophan that has this really big R group that sits there jams things up and it doesn't have as much flexibility. So there are limits. Okay? Now, proteins can be classified with 4 different levels of structure. The primary sequence is which amino acid follows the next. That can actually come from experimental conditions, which we'll talk about. We can actually use chemistry to derive the sequence. Okay? We can also use the DNA strand to predict what the primary sequence is. Knowing the primary sequence is fundamental for us to understand what the protein does. Okay. There is a second level called secondary structure. This is structure for amino acids very close to each other. How is the amino acid right next to it related in 3-dimensional space? We're talking about those twists. And then we're talking 3 and 4 away. Very close, there are certain patterns of structure that happens in amino acids that are very close to one another. And then there's another level of structure called tertiary structure. Tertiary structure is the overall fold of the protein. So it's its true 3-dimensional structure. Another way to say that is, how are the amino acids very far away from each other in the linear sequence? How are those related? Sometimes amino acid 1 and amino acid 333 sit right next to each other in 3-dimensional space, but in sequence they are very far apart. And so we get that context in the 3-dimensional structure. Usually the 3-dimensional structure is what causes function, but as we become more complex, the quaternary structure becomes a very important part in which one protein, polypeptide, one chain, and a second chain come together to create a more complex multi-subunit protein, and that's quaternary structure. How 2 chains interact amongst themselves. So we need to study a protein at all levels. Every protein has at least 3. It doesn't have to have 4. But when we talk about mammalian proteins, most of them do have quaternary structure. They do not work independently by themselves. There are experimental conditions or chemistries we can use to extract these different levels of information. And we'll learn those. So primary, the amino acid sequence once after the amino acid that begins and continues on all the way through. Secondary structure, spatial arrangement of amino acids near one another. Tertiary, spatial arrangements of amino acids far apart. Also, the disulfide linkages are classified in the tertiary structure. And then quaternary structure, more than one chain, or we usually call them subunits, will constitute. Subunits could be the same protein, so you have multiple things of the same working together, or you could have different chains that are working together. Ooh. How does that make you feel? I don't like that one. Okay, we're going to come back to it. We're going to use it in a little bit different way, the Henderson-Hasselbalch. Okay, what we're going to do is we're going to relate this. The numerical value of this and the numerical value of that, and then you're going to make a prediction of, is the structure you see more this, the deprotonated form, or is the structure more this, the protonated form? Okay? This has nothing to do with your calculator. Okay? But I'm going to give you some principles to be able to use this and predict, should it be protonated, should something be protonated, or should it not be protonated? Okay? And how these things interact in the buffering system. Okay? And so, all right, let me do this. I'm going to add a slide. Okay, there's a slide. Okay. Here is a normal protein. Okay. Very technical there. Okay, here's the protein again. Oh, sorry. Here's the protein again. All right. What usually ends is a C terminus. And what usually is at the N terminus is this. The protonation states are very, very important. The N terminus is always protonated. The C terminus is always deprotonated under normal pHs, which is about 7.4 in solution. Now, why is that? Okay. The pKa of the nitrogen for all of the amino acids is between 9 and 10. The pKa of all of the acids for every amino acid is somewhere between 2 and 3. Okay? The pH of the solution is about 7.4. Okay? So, this is how I remember it. If the pH is higher than the pKa. All right, so what does that mean? If the pH number, 7, is higher than the pKa, 2, then deprot. Take off the proton. If the pH is lower than the pKa, then protonated. That is really, can't even read that and I know what I'm writing. Stop. Numerical value, 7, lower than 9, protonated. must be at least 1 unit above or below. Okay. If the pH is equal to the pKa, then what? 1/2 of the sample is protonated and 1/2 deprotonated. Meaning that you never have one amino acid. They're so small, you never have it. You have a collection of them, or you never have just one protein. And so if the pKa is exactly at the pH, then half of them are in the salt form mathematically, and half of them are in the acid form. Or half of them are deprotonated, half of them are protonated. Yes.
So, do you mean if the pH is higher than the pK of the N-terminus?
Of anything. Of anything that you're looking at. And so in this case, if the pH is, which one? Higher than? Okay, so the pH here is higher than this one. Oh, say wait. The pH here, 7, is higher than this one. So this one has to be deprotonated. This one is not higher. That numerical value is not higher than that. So it can't be deprotonated. And the pH here is lower than this one, and it has to be protonated. That's why that one is protonated. But it has to be at least 1 unit value either up or down. So in this case, 7-ish is the normal pH. The 9 and 10, always protonated. The 2 and 3, always deprotonated. Okay. Now, that's all well and good for the C-terminus and the N-terminus of proteins, but there are all of these R groups. Each one of them may have a proton that can be deprotonated or not, and then once you find out if it is, everyone has a different pKa. And so you will have to make an adjustment for the N-terminus and the C-terminus, and then every R group that is available to you. Excuse me. Okay? So, normally the N-termini is always protonated, the C-termini is always deprotonated, and the R groups can have varying pKa's, of which they're in a chart somewhere, or they'll be in a chart that you will be given on the exam. I will not ask you to memorize individual pKa's of R groups. That's useless, because you can always look those up. But I do want you to understand that in general, the N-termini between 9 and 10, you can actually look it up and look at the real value. And the C-terminus between 2 and 3, you could look up those real values. But that gives you enough of a clue based upon a pH of 7 to know whether they're protonated or deprotonated. Okay? So, you did math without doing any math. Most people like that. Yeah. That's your favorite kind of math. Do a math with no math. All right. Let's take a look at this particular piece. This is the application of the pKa's in glycine. Glycine has 2 functional groups that can be protonated or deprotonated. Okay? The N-termini and the C-termini. And you can see that the N-termini is 9.6, that's between 9 and 10, and the C-terminus is 2.34, that's between 2 and 3. And so we can change the pH of the solution, okay, and show different states of this amino acid. And if you take a look at it, you can actually draw 3 versions of glycine. One in which it is positively charged with a positive charge of 1. One of which is neutral, even though it has a positive and a negative charge. You know what that's called? It's neutral but has positive and neutrals? Zwitterite. And then you can draw a structure in which it is negatively charged 1. But it all depends upon what pH the solution is in. You haven't changed what glycine is, you just change the conditions in which the glycine is seen in. Okay? Now, there is a zone where you have to mathematically calculate how much protonated and deprotonated are, and that's between 1.34 and 3.34. There's a distribution, okay? But you know the charge of, or you know the pH where this exists. What would be the pH that this one exists at? From what range to what range? This one right here, this first one. It only exists when the pH is between what and what? Between 0, that is correct. But how high can it be? 1 point, when you get up to 3, 4, that's where it's starting to either protonate or deprotonate, and that one happens at the acid. If you're at 1.34, you're right on the edge where everything is protonated. But once you move a little bit more basic, the solution, you're starting to deprotonate that. And between the 1.34 and the 3.34, there's a distribution, a percentage distribution. The only one that you can guess is when the pH is equal to the pKa. That is when you will have a full protonated amine, and what you would show is half of them are negative because half of them would be deprotonated and half of them would be neutral. So you don't get a full charge, you get half of the charge that's possible. All right, this one exists in a particular pH range. What would that be? Yeah, it would be 3.34 to 8.6. Okay? Now, technically, that is true. All right? There's a distribution. Now, that is absolutely neutral, correct? Okay? When it is absolutely neutral, okay, the pH is actually 5.97. Okay? Even though you're past 1 unit, okay, and we consider it, that's rounding. Okay? If you get to 99.8, we'll round that to 100% protonated or 100% deprotonated for, for grins. And so, government work will say that. The actual value called the PI value is when this becomes absolutely neutral. That means all of it is deprotonated, all of it's protonated, and that is the point at which it becomes absolutely neutral. So that's the real value. But for working knowledge between 3.34 and 8.6, we consider it in the middle state. What pH values define it into this last state? It has to be what pH value to what pH value? 10.6 all the way up to 14. All right. If I plop the glycine into the body, which of the three versions will exist? The first one, the second one, or the third one? The middle one? Middle one, right? The other ones theoretically can exist, but they're not going to exist in your body too often. Okay? The closest one that you might get to is the first one in the body. But where would you be? The stomach. Okay? When it's digesting. That would be a place in which the pH in your body gets way down into the 1 range. But that's under very special conditions. That's just not floating around. Okay? Where is your body pH of 14? Only if you're dead in the vat of base. Okay? That one doesn't exist naturally. Okay? Not a good place to be. All right. Now, one thing about this, glycine can be used as a buffer system in an experiment if you need. If you need to buffer around 2, you can do. If you need to buffer around 9.6, you can do that. And we do use glycine as a buffer system. Not only is it an amino acid, but it can be used as a buffer. Glycine is beautiful. It is very simple. You can actually produce that. It's 2 titrations in a row. You could actually produce that in the laboratory. Glycine is probably the best one of these. Okay, here's more like the real. Those are 3 titration points, and they're not so exact, right? Okay, especially, let's say, this guy right here. This one right here. What charge is this one right here? It's what? It's neutral. It's got a positive, got a negative, and the side chain is neutral, so that's neutral. Where would you find that one? Okay, the pH has to be at least 3.19, but it has to be lower than 4. Well, nope. Has to be lower than 3.25. There's a thin seam right there to get in there. Okay, because you're having protonation state. Now, this pKa is referring to what happens there. Okay? This pKa, oh, I'm sorry. I messed up. This one. This pKa is actually the deprotonation of that one. And this pKa is when you start deprotonating that. So each one of those pKa's refers to one different place in which this particular amino acid can be deprotonated. Okay? And so that one's not as nice and neat. That's not a titration I would give even a general Chem 2 student. Although your data might look like that when you actually do your experiment, I wouldn't give you that one. I would actually give you one that should look like that, where you got nice, and you might produce something that looks like that, okay, because technique, let's say, all right? The pKa of the histidine, and you can see where the histidine actually becomes protonated. It's right here, that nitrogen has a pKa of around 6, which is close to the pKa of the 7.4. It is a 1 unit off. Many times the protonation state depends upon if it's inside the protein. And so histidine has 4 different protonated, or different versions of the molecule itself. Okay? And so I could ask you as a test question, drawing this, I could draw this molecule and ask you which pH range would that exist in, as long as you have all the pKa's values given for the N-terminus, the C-terminus, and the R group. That's something I could conceivably ask you about. Or I could give you a pH of the solution, give you all of those, and ask you to draw out what version is it supposed to look like. Okay, so that's the kind of thing that I might ask you from what I presented to you.